Welcome back. So, we're talking about different types of vaccines and different types of characteristics. So, not all vaccines are going to work the exact same way. They can be made slightly differently, where they will have different effects on your cells and different types of immunity that they'll evoke. The first type are live attenuated vaccines. These are vaccines that are living, the pathogen is living, but it's been attenuated, it's been weakened. So because it is still living, it's able to multiply and mimic an actual infection, but because it is attenuated, it won't actually cause any disease symptoms with it. You may have things like a fever and pain just that signify your immune system is working, but no actual symptoms of an actual disease. Because they are mimicking an infection, because they're growing like a living thing, they will give you a very good immune response. You'll likely have both cellular and humoral immunity. So you'll have memory T cells and memory B cells both available in case your patient ever interacts that dangerous pathogen in the wild. Then there are inactivated killed vaccines. So these are killed, they are not living anymore. So because they are killed, they are safer, but they won't have a very strong immune response. You'll likely need to get repeated booster doses every 5 or 10 years. And mainly it will only be a humoral immune response because your B cells will be able to interact with it outside of a cell, but it won't actually infect a cell to get any sort of T cell response. Inactivated vaccines are going to be more appropriate when the pathogen is not as common. So if it's something that is more that a lot of people are going to be sick with, you might favor a live vaccine. Whereas if it's less common, less dangerous, that safer vaccine, the inactivated killed vaccine, would be more appropriate. So I think there were two different types of polio vaccines. And when I was a child, the live attenuated vaccine was what was given. So my polio vaccine was a little cup that I drank. However, there was some risk that polio vaccine could mutate into something pathogenic. And so they would say, if your baby's had a polio vaccine, don't change their diaper unless you've also been vaccinated against it because you have that slight risk. But now there is less risk of polio going around. And so that inactivated killed vaccine is what's used now. So if you took your child to the clinic now to get a polio vaccine, they'd have an injection in their arm of this killed vaccine. Next are subunit vaccines. These use just a subunit, just a fragment of the antigen. Sometimes they will be called recombinant vaccines, which are subunit vaccines produced by genetic recombination, recombinant. Toxoids, we've talked about toxoids. These are inactivated toxins. And virus-like particle vaccines that look like a virus on the outside, but they don't have any DNA or RNA. They don't have any genetic material inside. Polysaccharide vaccines are made from molecules in a pathogen's capsule, although they are not very immunogenic. And conjugated vaccines, which are used for diseases in children with a poor immune response. So they conjugate them. If you remember talking about conjugation in chapter 17, that conjugation will allow for a stronger immune response. Nucleic acid vaccines are going to use a nucleic acid. So they'll just inject a piece of DNA. That DNA will get into the cell, produce a protein. That protein will be displayed on your patient's cells and will stimulate both humoral and cellular immunity. And recombinant vector vaccines use avirulent viruses or bacteria that have been modified to deliver genes coding for antigens. Of the COVID vaccines that are currently available, there are mRNA vaccines, which would fall into the nucleic acid vaccine category, and there are some that use an avirulent virus. So there are some that are recombinant vector vaccines. There is some material about COVID vaccines provided in this week's class material, so feel free to go look at that to learn a little bit more about those COVID vaccines. Here's a diagram showing what DNA vaccines do. So the DNA is injected, the DNA gets into the cell, and your cells will start displaying the protein. Then your immune cells develop an immune response. So you have a memory response in case that was ever infecting your cells.
Vaccines may contain different versions of the pathogen. Depending on what they contain, these vaccines may be more or less effective in inducing long-term immunity. Two types of vaccine include the entire pathogen, attenuated and inactivated. In both types, the pathogen has been modified to be less virulent. The attenuated live vaccine contains the living pathogen, but this pathogen has been weakened so that it's unable to cause disease. But the pathogen can still reproduce. This type of vaccine has several advantages. It's the most effective at inducing lifelong immunity, and when the pathogen is a virus, it will induce both humoral and cell-mediated defenses. The disadvantages are that the pathogen can cause disease in an immunocompromised person and occasionally can revert to its virulent form and cause disease even in a healthy person. An example of an attenuated vaccine is the MMR, which protects against measles, mumps, and rubella. The inactivated whole agent vaccine contains killed pathogen. Inactivation makes this type of vaccine safer. There's no danger of causing disease, but the killed pathogen can't reproduce and often has weaker antigens, so it only stimulates humoral immunity and often requires additional booster doses to gain full immunity. The vaccine currently used for influenza is an example of this vaccine. Toxoid vaccines contain no pathogens, only inactivated toxins from a bacterium. These toxins are treated with chemicals or heat to make them less dangerous, but this also makes the toxin a weaker antigen. Therefore, toxoid vaccines only induce humoral immunity and require periodic booster vaccines to maintain immunity. An example of a toxoid vaccine is tetanus vaccination. Genetic sequencing and recombinant DNA technology have allowed immunologists to delete virulence genes from the pathogen, thus preventing an attenuated pathogen from mutating to a virulent form. Subunit vaccines, such as the one used for hepatitis B, contain only non-pathogenic antigens of the pathogen, not the entire cell or virus. Thus, they cannot cause disease. Conjugated vaccines, such as the one for Haemophilus influenzae B, take the capsule polysaccharides of a bacterial pathogen, which are not very antigenic, and combine them with a highly antigenic protein to gain a more potent immune response. DNA vaccines are currently being developed that contain only the pathogen's nucleic acid. Thus, the pathogen's antigens could be made by the host cells, and they could induce immunity without ever introducing the pathogen itself.
Okay. So sometimes also an adjuvant needs to be added to a vaccine. This will improve its effectiveness. There's only 2 approved adjuvants in the United States, alums and monophospholipid A. These are used when the vaccine on its own is not going to yield a very strong immune response. So the adjuvants will improve the innate immune response of that vaccine. So now you can answer these questions. Experience has shown that attenuated vaccines tend to be more effective than inactivated vaccines. Why would that be? What type of vaccine is an adenovirus that expresses a malaria CS protein? So one type of virus expressing a different organism's protein. Which type of vaccine is a live measles virus? Is that inactivated, attenuated, recombinant, or DNA? And what is the value of an adjuvant? Okay, I will come back and talk to you one more lecture video, a couple of issues and problems that have to be discussed and thought about when we're talking about vaccines. See you in a little bit. Bye.